Good morning, everybody. Welcome to our Shed Shares. For today, I'm going to introduce you all to Chris and Esther, who are going to take us through the Resilient Dairy program that we are working on here at LIC. So Christine is the research lead here at LIC, and Esther is one of our many scientists. So thank you, Chris and Esther, for joining us. I'm now going to hand you all over to Esther.
Good morning, everybody. So before we kick into the topic of today, which is around disease and wellbeing screening, I just wanted to take an opportunity to briefly talk about the Resilient Dairy program in general. So LIC is leading the Resilient Dairy Innovative Breeding for a Sustainable Dairy Future program. And this is a research program that is with investment and support from both MPI and Dairy NZ. It's a 7-year sustainable food and fibers futures investment program, and it was launched in June 2019 at the Field Days. So in short, Resilient Dairy seeks to utilize disease management technologies and genomic advancements to enhance the health and well-being of our national dairy herd and also improve our cow productivity. So there's a large amount of work that falls under the Resilient Dairy program. And just to give it a little bit of a structure, we describe them under two headers that you can see on the slide there. So the first one is animal wellbeing and health diagnostics, and the second one is genetic innovation and genomic advancement. And I will just briefly talk through all of the points of the projects that sit underneath that before we delve into the topic of today. So the topic of the day is the disease and wellbeing screening and diagnostics, as well as Chris will touch base on the biosecurity screening a little bit. So both of these projects, they're all around providing diagnostic tool suites really, and early identification of diseases into our dairy population. So part of this work includes leveraging the analytics of a herd test and developing new techniques to identify animal health traits such as pregnancy status or Johne's disease, for example. And then the biosecurity screening really is around providing early identification of notifiable diseases. And samples of both of these first two headers could either be milk, effluent, or feces. So Chris is really going to delve into this one. The third point on the animal wellbeing and health diagnostic there is facial eczema diagnostics. And this is all around an investigation into the potential of a milk-based diagnostic tool that can identify animals that in your herd have facial eczema. And if you're interested in the facial eczema topic, please have a look at the website, since we did a shed share on facial eczema a couple of months ago. So please have a look at that, and feel free to approach us if you want to have a chat about it. Then just moving on to the second big topic there, genetic innovation and genomic advancement. So the first header underneath there is genomic inbreeding, and what we're trying to achieve under this is, as the title says, is calculating inbreeding from genotypes. So it's about improving the accuracy of inbreeding in our population and into our animal evaluation model, which could increase the long-term health of our population, which is what Resilient Dairy is all about. Next one is genomic modeling, and this is developing an enhanced animal evaluation model that can incorporate new phenotypes as well as account for structural variants within the genome. And this is all about increasing the accuracy of the predictions that we're currently doing, which can then increase the rate of genetic gain. Third point, bullet point underneath there, is the incorporation of a New Zealand specific genome reference. And this is all around, as the title again says, in the past we have utilized, there was only one reference genome for cattle, and that was an American Hereford. So New Zealand has, with cooperation of others, has created a New Zealand breed-specific genomic reference. And this means that we can actually compare our genetic information to this specific reference. And this will improve the accuracy of genomic predictions and allow us to discover future genetic variations within our herd for the New Zealand dairy industry. So this is another very exciting piece of work underneath that kind of genetic and genomic advancements. Again, there we've got facial eczema. So this work leads on from the diagnostic one, and this is around developing a facial eczema breeding value. Again, please have a look at the previous shed share we did on facial eczema. And then the last two there are, the first one is sex chromosome investigation, and this is around determining the influence of the Y chromosome on key breeding areas such as male fertility, for example. So this is a really exciting and new piece of work that we're looking into. And then last but not least is Dairy NZ's info herds. So This is an investment from Dairy NZ, which goes into rebuilding the national evaluation system for dairy cattle to incorporate genomic information to facilitate, once again, faster rates of genetic gain. Before I hand over, I just wanted to make sure that have a look at the sheets here. We also have a header of Resilient Dairy underneath the LIC website, which you can find some information on the general kind of program that we run. And we also endeavour to use other forums like these JETSIAs, as well as the field days to present and explain some of the work that we're doing. And now I'll hand it over to Chris. Thank you very much.
Thank you, Esther. Thanks for giving a really nice overview of the whole Resilient Dairy program. So I'm just going to talk about a part of Resilient Dairy that comes under the animal health and wellbeing part of Resilient Dairy. And this is actually looking at dairy and New Zealand dairy farm microbiomes. Most of you probably have heard the word microbiome used in the popular press and in the media, usually in terms of the human gut microbiome and whether it can be used as a metric of how healthy or unhealthy somebody is, or if you actually can change the gut microbiome in humans to change things about people, such as whether they're fatter or skinnier. So we're taking this out onto dairy farms, and because of the herd testing LIC does, we've started by looking at the milk microbiome. So really, our main goals for this work is part of Resilient Dairy, so it really is centered around herd and animal health, and really for early disease detection. At the moment, the way diagnostics works is people will think that, oh, I have the sick cow, let me see if the sick cow is sick with this particular disease. And what we would like to do is actually move away from that and move to sort of early screening of all diseases simultaneously. So we know when a cow is going to get sick even before she gets sick, and we know exactly what she's going to get sick with, and which strain and which sort of resistance to antibiotics she will have. So animals can be treated early and can be treated right the first time with the right drug. This is all about animal well-being. When we're looking at the microbiome, we don't just find things that are able to make the cow sick. We actually see things that might make the cow healthier, probiotics, and also things that may infect the farmers, such as leptospirosis. One of the great things about this program is that we don't actually need to know what it is exactly that we're looking for. Because there are some big databases in the world of where there's DNA sequence for all sorts of species, we can actually compare our data to those databases and look to find any new incursions in New Zealand. So being able to find new species entering New Zealand has some really big benefits. If you think about M. bovis coming into New Zealand, if we'd actually been able to detect its presence here earlier, it would have caused a lot less disruption to the New Zealand dairy industry. So this is something we've been talking to MPI about and how we might facilitate this. Now, it's mostly about the cow health, and part of cow health is actually the cow herself, and looking at those genotypes. And we can actually get cow genotypes as part of this work, just as an added bonus, pretty much as a freebie bonus. And these genotypes can then be fed into that genomic modeling and the genomic selection and other parts of the Resilient Dairy program. So what are we doing with milk, and why can we do it? Well, you might think that milk is just a liquid and it's just milk, but actually in the milk that comes out of cows, and especially in the herd test samples, there are lots of cells in there. There are cells that come from the cow, and you'll be familiar with these. These are what you get reported back as somatic cell count. But in addition to the cells from the cows, there are also cells from bacteria. There are lots of bacteria present in the milk. Some of these come from in the udder, some of these will come from the skin of the cow, and some of these will come from the environment that the cow is being milked in. There are also viruses in milk, and there are these really cool little thing called phage. And phage are actually viruses that infect bacteria. So sometimes if these are present, they're actually killing the bacteria, which I think is pretty cool. And we can look at all of these species all in one hit just by DNA sequencing. DNA sequencing technologies have come a really long way in the last 10 years. And if we extract the DNA from milk samples, we can actually just put that DNA onto a sequencer and we can see what sort of species are present. Just to give you an idea of that, DNA sequences come in all different sizes and shapes. These are 3 different types of sequences that the LIC has. This here is a little benchtop sequencer. It doesn't produce so much sequence, but it can do things quickly and cheaply. The middle picture here, this is sort of the big daddy of DNA sequences. This particular machine here can produce 6 terabytes of data every 2 to 3 days. So that's a lot of DNA sequence that we can generate very rapidly. And here we have quite a portable little sequencer. It doesn't produce the large volumes of sequence, but it can read really long strands of DNA sequence, which is quite useful to us. So we're going to use all 3 of these different types of sequencing to have a look to see what the milk microbiome looks like on New Zealand dairy farms. We've started mostly with herd test samples because we have easy access to herd test samples. And we've been working with 5 farms, the LIC farm called Innovation Farm, and 4 other farmers that we invited to be part of our pilot trial. We've also had those farmers send us bulk milk samples on the days that they've been herd testing. And we have 2 approaches that we're going to look for all the different species. One, which we're hoping will be a cheap and cheerful strategy, targeted sequencing, where we picked like our 50 or 60 species that are of most interest. And we worked with a number of vets to come up with a list of 60 species that we're most interested in looking at on the dairy farms, both in milk and in feces, and pretty much any other sample type that we can get our hands on. The other approach is just to sequence everything. This is a little bit more costly, but this is the approach that actually allows us to detect things we don't even know that we need to be looking for. So we've done some pilot trials using both of these approaches, and both of these approaches produce large amounts of data. I already said we can produce terabytes of data in just a handful of days. And I think we probably produced something in the order of 50 terabytes of data just in the pilot trial. But DNA sequence data in itself isn't that much use. We can say who's there, but we really need to find out how much of each species is there as well. And so we've done a lot of work trying to convert our raw sequence data into metrics that will actually be useful on farm. And this isn't trivial. Give us a call or send us an email and we'll share more about how we're actually doing it. But we transform our sequence data into something that's useful on farm. And now I'd just like to move to what we're actually using that data for. So the first thing I said that the somatic cells in the milk give us a lot of DNA sequence data from the cows. So actually from this herd test sample here, even with no ID, as long as that cow has undergone parentage testing, we know who that sample came from. We know which cow gave that herd test bottle with no label on there at all. And often we can actually say who was milked in the bale before she was, just because there's a little bit of milk that gets left in the lines. And this is quite useful for us to make sure we do actually have the samples that we think we have. But actually we're testing it out now to see whether the genotypes that we get from that DNA sequence can actually be used in genomic selection, because the more animals we have genotypes for, the more accurate our genomic predictions and our genomic selection can be. And if we can tell who gave us the sample, then one day we may be able to use this to help us with tracing where animals actually are. If we move away from the cow now and look at the non-cow species that we've seen, on the left we have this word cloud of these are just some of the species that we've seen in there, and the size of the words are, the size of the species names correspond to how often we see DNA sequences from them. Now, Staphylococcus aureus, this is a little bit inflated in size, this one, because we did actually start by picking quite a number of cows that we knew had Staph aureus infections, because we wanted to make sure that we could detect it. But for every herd test sample that we sequence, we literally see hundreds of species per sample. Now, some of these are coming from inside the udder. It used to be thought that milk was a sterile liquid when it came out of the udder, but in the last probably 5 years or so, that is no longer thought to be the case. There are definitely species coming from inside the udder. We see a lot of species that look like they're coming from the actual skin of the udder. But depending on farm, on the farms that we look at, we actually also see plenty of species that seem to be originating from soil or silage. whether or not the cows are lying down in particular areas and picking up bacteria from soil or silage that they might be eating. We're not quite sure about that yet. We see species that look like they're coming from the actual milking plant itself. This is particularly the case when we look at bat milk samples. Now, I've started talking and I've mentioned sort of mastitis type bacteria quite a number of times already, but in this project, we're not just looking at species that cause mastitis. We're looking for species that cause all sorts of diseases in cows, from not that aren't in the udder. So if a cow has a blood-borne disease, then we think that we should be able to pick that up. And I'd now like to show you just a little bit of data suggesting that we're right, that our hypothesis about picking up species that are not causing a problem in the udder is correct. So this is just a little bit of data from one of our early pilot trials. In this picture, every row is either a herd test sample, a bulk milk sample, or an effluent sample. And every column represents one of the species that we're looking at. This is only a very small snapshot of the different species that we've seen. And in here, where we see white, that means we don't see that species at all. And you can see none of the samples that we tested have M. bovis, and we have never seen N. bovis in the samples that we've been looking at. When we move to the blue, the darker the blue, the more of that species that's present. When there's red, that means we've seen a lot of that species in that sample. So there are a few of the usual suspects, Staphylococcus aureus and Streptococcus tuberous, that cause mastitis. And we do see these mastitis causing bacteria in some of our samples that we're looking at. But we also see Theileria. And for those of you that live in the North Island, you'll likely be familiar with Theileria. It's actually a protist, so it's a much bigger species than a bacteria, but it's a protist that's carried in ticks. And when the ticks bite the cows, this is actually a blood infection. And we can quite clearly see Theileria in these two cows here. Now, these two cows, they happen to be Innovation Farm cows. And when we called up the farm manager, he actually informed us that these two cows had been diagnosed with Theileria around the time that these samples were taken. And they've been treated and they're perfectly all right. But that gives us a lot of hope that by looking at the microbiome of the milk, we'll actually be able to make some conclusions about health. We put in this effluent sample because we knew that it was positive for Johne's disease. And this is the bacteria responsible for Johne's disease, and we detect that quite nicely. So this is all looking very promising in that we can detect all sorts of different things that might be useful to us in milk. One of the big questions we have to ask ourselves is how accurate is our measurement? Because the data would be worthless if we couldn't really measure things accurately. And there are so many species that we see, and it's very hard to know if we're getting the right answer or not. But for Staphylococcus aureus, we actually have a gold standard test that we can compare our sequencing-based results to. If anybody's done the Staph aureus testing that LIC commercially does, you'll be familiar with this test, that we report high positive samples. We report if samples are high positive, if they're detected, if they're suspect, and these green samples all represent samples for which the PCR test has not been able to detect Staph aureus. So when we actually compare the measurements from both of these different techniques, and these are very, very different ways of measuring the same bacteria. We measured the same bacteria in samples using the two different techniques. And we found where our PCR test was picking up Staphylococcus aureus, our sequencing-based approach was giving a near perfectly similar result, which is really, really promising. This stack of green samples up along the y-axis here, these represent samples where the PCR test hasn't detected Staphylococcus aureus, and where our sequence-based approach actually really does seem to pick up Staphylococcus aureus. So we're doing a little bit of work at the moment to try and prove or disprove whether or not our sequencing-based approach might actually be a much more sensitive way of detecting bacteria, and we really hope that it is. Now, remember, this is just one particular species out of the hundreds of different species in every herd test. And if we had commercial tests for all those other species, we could do a similar sort of a comparison. But at this point in time, we're pretty limited in the number of species that we're able to detect either and compare the same samples in the two different techniques. So we're thinking that this approach has a great deal of promise. Just to give you a bit of a scale of where we're at to date in the 2 years that we've been working on this, we have more than 5,000 milk samples that we've sequenced. These are from 5 farms where we've sequenced the same cows time and time again, herd test after herd test. At Innovation Farm, the LIC farm, we've actually done herd tests every month. And what we've been able to do is to track how the microbiome in the milk changes from month to month as the season changes, as the cows are being fed different things. There's a clear shift in terms of which species are present and how much of them is present. In total, from all these samples, we've actually identified 4,000 different species observed. So that's quite a lot. Many of these, nobody in the world knows what they do, but there are quite a lot of them, including the usual suspects that are known to cause problems on farms that we do see. So what's next? This work is progressing really nicely, and now that we've sort of got this pilot trial underway and finished around the herd test samples, what we want to do is we want to expand out wider into New Zealand. We want to survey what the milk microbiome looks like across New Zealand and see whether or not there are regional differences, or if every different farm is different, or the types of systems that farms are running, is that making a difference to the microbiome on these farms. And we want to do this because if we want to use this for monitoring health and early detection of disease, we need to know what the baseline is, what's normal on farms in this region or using this system of feeding. What do they look like, and just what species are present on farms in New Zealand, because nobody really knows the complete answer to that yet. So we are looking for farmers who are interested in being part of this really exciting study. So if anybody's interested in supplying us with some bulk tank samples in the coming season, we would really be very happy to hear from you. In this work, from both the individual herd test samples that we have and these bulk milk samples, what we're looking to do is to really screen what species are present in New Zealand dairy farms across the whole country, across farms of different systems, baselines of what's normal, what's usually there, how much is usually there, because this is our starting point for if we want to do that early screening and alert people when cows might be getting sick. So if anybody is very interested in supplying us with some bulk tank samples next season, we would really love to hear from you. There's an email address on this slide that you could email to say you're interested, or just to let us know what you think we should be doing. How much of a species is too much? If you have 1 or 2 Staphylococcus aureus bacteria, that probably doesn't mean you're sick. So how many is too many? And conversely, if with the probiotics, how many do you need to see before you're actually getting a positive effect? We're also really interested in the New Zealand strains of the different species. We often talk about Staphylococcus aureus as this one type of bacteria, but we would never talk about all cows being the same. And the same is true for any bacteria. There are all sorts of different strains, and some of these are more infective. Some of them have antibiotics resistance genes. Some will cause worse disease. So we're having a look to see what sort of bacterial strains we have in New Zealand for some of the common pathogens on New Zealand farms. And we really want to get a handle on where the different species that we're seeing are coming from, the ones that are coming from inside the udder versus on the skin of the cow. And this is quite important. Initially, when we were doing this work, we thought that actually maybe those species that weren't coming from inside the udder would just be noise and make our analysis difficult. But what we're seeing is that it actually gives us an insight to what's going on the farm, and that could alert people to what they need to be careful of. If there's a lot of a particular bacteria on the skin of an udder, it has a much greater chance of getting into the udder and causing problems. So these are some of the questions that we asked ourselves every day, and there are many others that we're working on that we can now begin to answer from the types of data sets that we've been generating from the last 2 years. But ultimately, it's really all about how can we use milk samples, bulk milk and herd test samples, to monitor the health of your herd and individual cows. So it's a great project. We've got a lot of work to do, but we're already making great progress. So if anybody has any questions, I'd be happy to answer them now. Questions or suggestions. We're always open to hearing from people to tell us what they would like to see us doing.
That's awesome. Thanks, Chris. We do have one question here. What does the research tell us on drinking raw milk? What do we know about that?
We haven't looked at that, because one of the things we aren't able to do yet, we may be able to do it in the future, is that from this, we don't say whether a bacterial species is alive or dead. So if it's dead, we see it, but we don't know whether it's alive or dead. So from our data at the moment, we can't really interrogate that. I think there are plenty of websites that have a lot of information on the species that are present in raw milk. There are a lot. It's again, how much is too much of any given species, and I know that MPI does have some strict rules around producing raw milk to try and minimise problems.
Brilliant. So we do have our herd test milk samples and parentage testing. What does that look like for the future?
We don't currently do that. We currently do parentage testing from ear punches. Something that I would like to see happen is us to be able to do parentage testing from milk samples. One of the challenges that we need to overcome there, and it is something that we're looking at and working on, is that contamination from one cow to the next. When you take the cups off of one cow, there's always a little bit of milk left in the line. And so when you put it on the next cow, the herd test sample is always a little bit of a mix. We hope that one day we can actually bioinformatically separate out which is coming from which cow. So we're working on that.
Awesome. I had a question there for you too, Chris. Really nice job. You answered a lot of my questions during your talk. My question was around, you kind of touched base on we need to get a profile to understand how healthy our cows are, but are we comparing it at all with, say, overseas barn-based cows, or like even other dairy products like goat and sheep microbiomes?
We aren't at the moment, but those are some good ideas that we should do. It's really hard with the question of what is a healthy cow is really hard. You know, with people go to the doctor and they say, oh, I have a terrible headache or something, but our cows are really stoic and they won't show us that they're sick until they're really quite sick. So we're actually hoping to do some work with some of the new collars, farms where there are some collars that can monitor animal health and look at the microbiome in association with the metrics that those collars are recording. But actually, one day comparing with overseas farms would be a fantastic idea.
Yeah, I think my question really came around the marketing side of it as well. Like, could we somehow utilize some of this information and showing, that dairy products, are healthy, and yes, there's stuff in there, but there's stuff in everything.
Yeah, hopefully we can. We're really the first people doing this on a large scale. So at the moment there isn't data to compare with from other countries. But I think with DNA sequencing costs coming down all the time, that hopefully soon, or hopefully when we show this science to the rest of the world, people will think, oh, we need to be getting on that bandwagon and having a look at that too.
Super. Thank you so much, Chris. That was a really good overview. And of course, Esther, your insight into the project itself was very insightful.